Animal cloning.
The cloning process. The, the cloning process usually involves removing the genetic information known as DNA from a cell taken from one animal and placing this into an unfertilized egg that has had its own DNA removed. This egg is then artificially stimulated to start developing into an embryo and placed into a surrogate mother who will eventually give birth. What it's used for. Cloning techniques are used to try and produce exact copies of a particular animal, which might still be alive or may have already died. The use of cloning technology is often necessary by statements that the research could lead to cheaper methods of producing food products.
History. In the early 1900s, Hans Driesch, a scientist, cloned two sea urchins by getting a two-celled embryo and shaking it in a beaker until it separated. Each embryo then grew into an adult separate e-urchin.
Dolly. The most successful in the field of animal cloning was seen when the cloned sheep Dolly was created in 1996, which could survive and also naturally reproduce itself. Unlike the previous animal cloning attempts, Dolly was not created through a developing embryonic cell. Rather, a developed mammary gland cell taken from a full-grown sheep was used to produce the cloned Dolly. Since the successful creation of Dolly, scientists have also succeeded in cloning many other animals such as rats, cats, horses, pigs, and deer.
Advancements. Gaber Vacha of Danish Institute of Agricultural Sciences created a technique where scientists slice the egg in half, discard unwanted genetic material, add new genetic material, and then fuse the halves together.
Advantages. Some advantages make it possible to create identical copies of the best individuals in a population, such as highest milk-producing cow or the largest hog. It also makes it possible to produce a genetically identical copy of an individual for an organ transplantation. Extinct or endangered animals can be repopulated through cloning. Disadvantages. A few disadvantages are there's a significant likelihood that the somatic cell used in cloning contains mutations which may weaken the cloned individual or have lethal effects. Another one is the DNA in the mitochondria of the donor egg differs from the DNA in the mitochondria of the somatic cell, which may cause nuclear mitochondrial incompatibilities that lead to death. Many, even thousands of eggs may be required to generate one viable clone, a wasteful, expensive process. Cloning reduces genetic diversity by producing nearly identical copies of somatic cell donors.
Ethical issues. Animal cloning often causes animal suffering, and animals are prone to develop severe health problems and abnormalities. Cloned animals also generally live very short lives, and the surrogate mother often has a painful birth, and the child usually dies after birth. How it's used. Animal cloning can be used to produce replicas of animals with the highest economic value to increase profits for business. Animal cloning can also be used for medical purposes, such as cloning animals for models for disease research and also stem cell research. Animal cloning can even bring back extinct species or help regenerate endangered species. Interesting story is that a family in USA paid the University of Texas $2.3 million to have their dog Missy cloned. Wouldn't it be weird to have dogs cloned in the future? Yeah.